Hi everyone, my name is Leonardo Rivera, and today we'll be talking about associations in Alzheimer's disease between intracranial vascular metrics from MRI and beta amyloid and tau from PET. Cerebrovascular disease has been strongly linked to dementia due to AD. Current hypotheses suggest that CVD is present in prodromal Alzheimer's disease and may be involved in the evolution of AD. This means that AD-CVD hypotheses need testing to determine what kind of interactions are happening. One of the major limitations to study these questions is the lack of sensitive markers to characterize CVD. However, with recent advances in quantitative MRI, functional vascular imaging is possible, including flow imaging known as 4D flow MRI. Using imaging methods pioneered at UW-Madison, with 4D flow MRI, we can measure blood flow velocities in the intracranial arteries and veins with whole brain coverage, high spatial and temporal resolution in around 6 minutes. And here you can see an example of a 4D flow scan displaying the blood flow velocities along the circle of Willis. In this study, we characterized cerebrovascular health using this technique in a group of AD biomarker confirmed subjects. So a total of 136 subjects were recruited from the ADRC and RAP studies. 4 groups were established, including controls, amyloid positive, amyloid and tau positive, and amyloid and tau positive, but also cognitively impaired subjects. Amyloid and tau positivity was determined from PET imaging. Importantly, no significant differences were observed in traditional cardiovascular biomarkers, including blood pressure, heart rate, and white matter hyperintensities. Initial results showed total cerebral blood flow was independent of AD biomarker status once adjusted for brain volume. One thing that we can measure with 4D flow is the dynamic evolution of the cardiac pulse wave. For example, the pumping action of the heart generates a pulse wave, and this pulse wave travels through the arteries, the intracranial arteries, reaching the capillary bed and returns to the heart through the venous system. By characterizing the cardiac pulse wave as it travels from the intracranial arteries, the brain parenchyma, and finally the veins, we can establish the transcapillary pulse wave delay transit time. And these transit times are indicators of both pulse wave transmission and vascular compliance of the macro and micro circulation. In our study, we found that shorter transit times were correlated with AD biomarkers, but no major differences were observed after an amyloid positivity status was reached. Here you can see transit times for 4 groups measured in the internal carotid artery and the basilar artery. We can also study beat-to-beat blood flow fluctuations. In this example, you can see blood flow changes in the superoccipital sinus, that is a large intracranial draining vessel. From these heart beat-to-beat fluctuations, we can derive low-frequency oscillations, which are a surrogate marker for autoregulation-related vasomotion and respiratory dynamics. And here you can see LFO's power spectral density analysis. On the left, we see LFOs in the arteries, on the right, on the veins. And LFOs between controls and amyloid positive subjects were similar. However, there was a significant reduction of LFO content in subjects that were both amyloid and tau positive. To summarize, we can go beyond traditional and structural markers of cerebrovascular disease to more specific markers of vascular health. For example, the shorter transcapillary pulse wave delay transit times observed in amyloid positive subjects might suggest arterial and capillary stiffening, while the observed diminished LFO content in the amyloid and tau positive subjects might be related to intracranial vessel wall modifications, including changes to the smooth muscle cells, which are thought to drive low-frequency oscillation vasomotion. And finally, we need to determine if these cerebrovascular disease markers can help improve modeling of AD neuropathological trajectories, which then can help us identify cerebrovascular health therapeutic targets. And with that, I would like to thank you for your attention.